Hi, my dear students. Myself, Anu Shashidharan, Academic Director and Biology Faculty of Rankings Entrance Training Academy. I welcome all of you in my new video lesson. Today, I am going to talk about one of the most important chapter of class 10. How do organisms reproduce? Part 3. Dear students, today we are going to discuss about vegetative propagation. As you know, vegetative propagation is an asexual method of reproduction. Do you know what is the meaning of asexual reproduction? Yes, in this type of reproduction, the new organisms produced from a single parent without the involvement of gametes or without the fusion of gametes. Vegetative propagation is an asexual method of reproduction. Dear students, in this method, new plants are produced from parts of the mother plant such as roots, stem or leaves without the help of any reproductive organs. When you consider the green plants, the flower is the reproductive organ or reproductive world. And all other regions, the root, stem and the leaves together called the vegetative parts. Yes. So, in case of vegetative propagation, the new plants are produced from the vegetative parts of the plants, especially root, stem and the leaves. For example, the sweet potato and dahlia developed from the root of the parent plant. Okay, some plants develop from the stem of the parent plant. For example, potato, banana, etc. In case of Bryophyllum, as you know, the new plants produced from the leaves or stem. Dear students, there are two different types of vegetative propagation. First one, natural vegetative propagation, and second one is the artificial vegetative propagation. Now, we are going to discuss in detail about natural and artificial vegetative propagation. First of all, let me discuss about natural vegetative propagation. Dear students, the plants may propagate naturally by way of their stem, root or leaves. We can collectively call the natural vegetative propagation. When you observe in nature, you can see that various plants, they can produce their young ones from their vegetative parts, especially root, stem and leaves. Now we are going to discuss about certain examples. First of all, in case of Bryophyllum, the buds are present on the stems as well as on the leaves, which can develop into new plants. First of all, look at this diagrammatic representation. You can see that special types of buds are present along the margins of the leaf. This is the Bryophyllum plant. You please carefully observe this diagrammatic representation. You can see that so many special types of buds are seen throughout the margins of the leaves. These buds may get detached from the leaves and grow to produce a new Bryophyllum plant. Okay. Next one, in case of money plants. Do you know money plant? Yes. This is actually the money plant. This money plant propagate by using their stem. You know, if a piece of stem is dipped in water, what happens? The new roots will appear after a few days. Look at the second diagrammatic representation. Here you can see some stem of the money plant have already been dipped in water. After a few days, what happened? The leaves may develop on the stem. Later, each and every piece of the stem will gradually grow into a new money plant. Is it clear? We have to discuss two more examples. Example 3, in case of some tubers like potato. Did you see the potato? Yes, if you carefully observe the potato, you can see that it has a number of buds on it. It is called the eyes of potato. Each and every bud on the tuber grows into a new plant. Okay, last one, in case of sweet potato and dahlia, you can see that the new plants are propagated, the new plants are developed from the roots, especially the root tubers. Is it clear? In sweet potato and dahlia, they are propagated by using their root tubers. Is it clear? Next one, we have to discuss in detail about artificial vegetative propagation. Do you know what is the meaning of artificial vegetative propagation? Yes, the process of growing many plants from one plant by man-made methods is called artificial propagation of plants. Dear students, the artificial propagation includes three important methods. First one is the cutting, second one layering, and third one grafting. We have to discuss in detail three different types of artificial vegetative propagation. First of all, let me discuss about the cutting process. Dear students, in this method, the parent plant having some buds on its stem is cut and its lower part is buried in the moist soil. After a few days, you can see that it gives rise to the roots and shoots. Thus, a new plant is formed. For example, you are very familiar in case of rose plant and sugarcane, the major method of propagation is stem cutting. I hope you might seen the stem cutting method for the propagation of rose plants in your house. Second method is called layering. Okay, you just first of all go through this diagrammatic representation. What is the meaning of layering? Layering means when the roots are artificially induced on the stem branches before they are detached from the parent plant for propagation is known as layering. For example, in this method, you can see that the branch of the plant, can you see this branch of the plant is pulled towards the ground and is covered with the moist soil, leaving the tip of the branch exposed above the ground. Can you see? Here in this diagrammatic representation, we had selected two branches and a part of the branch we have pulled towards the ground, especially in the moist soil. Okay, you are very familiar when the part of a plant, especially the stem, come in contact with the soil, what happens? They may produce the roots, isn't it? So you just imagine whenever the plant start its growth, you can detach the plant, I mean this part from the parent plant, and we can use this as a new plant. This is the method we can collectively call the layering methods. So in this diagrammatic representation, you can see that from this mother plant or from this parent plant, we can produce two plantlets. Yes. Third one is called grafting. Have you heard about grafting? Yes. It's a method in which the stem cut of two different plants are joined together in such a way that the two stems join and grow as a single plant. So this method we are commonly using for incorporating the characteristic features of two different plants. Here you can see that the stem with the root is called a stalk and the other is called a scion. In this diagrammatic representation, you can see that the stem of a plant which is having roots, this is called the stalk region, and the other parts we can call it as the scion. So what we are doing, the stalk and scion are arranged in such a way that in attached form. Okay, and afterwards we have to wrap around it and wax it. After a few days, what we can see, the new plants formed from this grafted region. That means the scion will start its growth and develop into a new plant. Can you predict the speciality of this particular plant? Yes. This plant may produce or this plant may have the characteristic features of two different plants. That means the stalk and the scion. The new plant formed by this method will have the characteristics of both the plants. This type of artificial vegetative propagation we can collectively call the grafting. Dear students, now we have to discuss about the advantages of vegetative propagation. As you know, the plants produced by this method will be exactly like the parent plants. Why? Because this is a type of asexual reproduction. There will be no gametic fusion. That means there will be no variation. All the young ones are the exact copies of their parents. Second one, many plants can be produced from only one parent. So clear? The fruit trees grown from cuttings or by layering we have learned they can start to bear fruits much earlier. Okay, it is an easy and cheaper and quicker method of propagation. The plant produced by artificial method require less care in their early years. Many seedless plants can be produced. It's the most important point. I hope you might see different types of seedless plants. In case of seedless plants, we are not able to produce the young ones with the help of the seed. So in such type of plants, the only one method of propagation is artificial vegetative propagation. It is helpful in propagation of ornamental plants. These are the advantages of vegetative propagation. Dear students, we have to discuss one more concept under the asexual reproduction that is called tissue culture. I hope you might hear about tissue culture, isn't it? Yes. The method of propagation which helps to obtain a large number of plantlets from a small piece of tissues taken from any part of the plant body. You just carefully go through this diagrammatic representation. Suppose you think about we need to produce so many young ones from this parent plant. So what we have to do? First of all, we have to select a small part of the plant for this type of propagation. The part of the plant we are using for tissue culture is called explant. In most cases, we are using the shoot tip as the explant. Is it clear? Dear students, this method of reproduction we can collectively call the micropropagation. Why? Because you are very familiar, the small size of the plant material is used for this method. I said the explant, it may be the shoot tip or root tip or even a small piece of the leaves. So clear. That is why this type of propagation we can collectively call the micropropagation. Dear students, you can see that the different steps in tissue culture. In this method, first of all, the explant is cultured on a medium containing necessary hormones and nutrients. This is the explant. Explant means the small part of the plant we are using for micropropagation. First of all, we have to inoculate the explants in a particular medium containing necessary hormones and nutrients. This is actually a growth medium. Okay, as a result, what happened? This result in the formation of an undifferentiated mass of cells called callus. Dear students, you just imagine this group of cells. All the cells arise from the explant. So all the cells are similar to that of the parent plant. Okay, this group of undifferentiated cells we can collectively call the callus. Dear students, this callus then transferred to another medium for differentiation in the plantlets. Okay, this callus again cut into pieces and that should be enter or that should be transferred to another medium which having rooting and shooting hormone. Because of the presence of rooting and shooting hormone, what happened that leads to the production of root and shoot from the particular tissues. As a result, that should be converted into a fully developed plantlet. Dear students, these plantlets we are protecting in the laboratory conditions. Okay. Afterwards, we have to transfer to the greenhouse up to and plant that should be grown in the greenhouse up to its maturity. Whenever the plant becomes mature or whenever the plant should attain a particular size, we can transfer to the normal field for cultivation. This is actually the method of tissue culture. So in the second diagrammatic representation, you can see so many plantlets are actually developing in a tissue culture laboratory. Now we have to discuss about the advantages or applications of plant tissue culture. Dear students, the tissue culture is a type of asexual method of propagation which are having so many advantages. We are going to discuss one by one. It helps in the rapid multiplication of plants. Dear students, we can produce thousands and lakhs of plants from a single mother plant. A large number of plantlets are obtained within a short period. Plants are obtained throughout the year under controlled conditions independent of season. Why? Because all the processes of tissue culture can be done inside the specially designed laboratory conditions. It is an easy, safe and economical method for plant propagation. In case of ornamentals, the tissue culture plants give better growth, more flowers, and less fallout. Dear students, genetically similar plants are formed by this method. As you know, this is a type of asexual method. We are using a small part of a single parent plant. That means all the plants developed from this method are exactly similar to that of the parent plant from which we have selected the explant. Dear students, the rare plants and species are multiplied by this method and such plants are saved. All these are the different applications or advantages of tissue culture. Dear students, I hope you have understood all these concepts thoroughly. I request all of you have to go through this video lesson again and again for better understanding. So we will meet next day with another video. Thank you. Have a nice